So let's now consider how evolution can be described as changes in allele frequencies. This is obviously a way that we describe evolution now that was not in a way that evolution was described in the 1800s. But this is a particularly useful way of describing evolution as we describe the different kinds of mechanisms that can cause evolution. I think that will be more obvious as we walk through some examples. So one of the important things to note as well about evolution is that populations and species, when one says that populations and species evolve, this means that their characteristics change through time, and specifically characteristics that have a heritable basis. But one can define evolution as changes in allele frequencies over time. And I've got a very simple example for you right now. This group of ovals here is supposed to represent a group of alleles in a population. And so let's now go through a couple of definitions to make sure that you understand what's meant by particular words. And of course, anytime, by the way, if you're reading in your textbook, if you ever hit a word you don't know, please look it up in the index or Google it, because it's really important that you understand all the words as you're reading the textbook, or you're not understanding the meaning. Anyway, a population is defined as a group of individuals of the same species that are living in the same geographic area at the same time. And so part of the significance of this is that these are individuals that can potentially interbreed, for example. And what is a species? A species is a distinct, identifiable group of populations whose members can interbreed. And we'll spend some more time talking in later lectures about species definitions, but this can be our working definition for right now. Now let's assume that we're talking about diploid individuals, so each individual has 2 copies of each gene, and let's consider something really simple, which is just we're going to consider one gene. So if we have a very small population, here's our little tiny population, 1, 2, 3, 4, 5. So in this particular population, we only have 5 individuals, 5 diploid individuals. So this would then be just 10 copies of the gene, or 10 alleles. And so let's consider this our starting point. So each one of these ovals then represents one allele in the population. And we're not even worrying about which pair of alleles are in which individual. We've just gotten rid of the individuals, so this is a highly conceptualized, simplified way of thinking about the alleles in a population. Now at this particular point in time in the parental population, 9 out of the 10 are blue and 1 is red. So we could say 90% of the alleles are of this blue type and 10% are of this red type. So the individuals in this little tiny population reproduce, they have offspring. Here is the next generation shown diagrammatically down here using the same representation where each single oval is one allele. We still, to make things really simple, we only have 5 individuals in the next generation as well. So we have a total of 10 alleles. Now notice, we now have 2 alleles that are red and 8 that are blue. So we actually have evolution going from this generation here to this generation down here. We've had a change in the allele frequency, and therefore, by definition, we have just seen an evolutionary change. This may be pretty puny and maybe underwhelming in this particular example, but this is an example of an evolutionary change. An evolutionary change doesn't have to be some huge thing that is accumulated over a very large number of generations. Now, to relate this back to what we were talking about earlier, which is natural selection, one has natural selection when individuals with certain alleles produce the most offspring in a population. So if this was an example of natural selection, then we can assume that individuals with the allele, which is shown in red here, have some advantage relative to the individuals that did not, which is why we're ending up with greater representation of that particular allele in the subsequent generations. So we'll return to that topic in a later lecture. But for right now, I want to emphasize the point that selection acts at the level of an individual, meaning that an individual lives or dies or has offspring or not, or more offspring or fewer offspring. But when one is evaluating evolutionary change, one is looking at the level of the population, not at the level of the individual, because the individual does not change in its allele frequency. The individual is unchanging genetically. Where one sees an evolutionary change is at the level of a population. So again, to go back to the example we were just discussing, because we saw a change in the allele frequency in this hypothetical population, then we did see an evolutionary change in the population as a whole, even though any individual did not undergo a change. That's a really important concept for you to wrap your mind around. Now, as these changes accumulate, of course, over very long periods of time, these changes can be more dramatic, such as what we saw in the pigeons, or what we saw in selection for Brussels sprouts versus cabbage, for example. And another really important point about how changes accumulate during evolution is to note that evolution is not progressive in the sense that it always leads in one particular direction in all branches at the same time. So if one considers figure 2519, for example, which is in front of you right now from your textbook, it shows the relationship, the evolutionary relationships between a couple of representative animals and plants and prokaryotes, and even a fungus in there as well. And over time then, we end up with a branching network of relationships, a branching network of different kinds of taxa. This is not a linear progression in living organisms as a whole. There may be a linear progression in any given non-branching part of a lineage. One can go forward or backwards in time and look at changes in a specific period of time, but in all these different cases, natural selection has led to a better match between traits and the environment. But notice that all these different organisms are ending up with different phenotypes and with different matches to their environment. The environment of a tapeworm is completely different from the environment of a vertebrate such as ourselves, which is completely different from the environment of a fungus. And so the kinds of traits that are advantageous for a tapeworm are different from the kinds of traits that are advantageous for a fungus. And so natural selection tends to shape different kinds of organisms in very different ways because the environment is very different. So this is what is meant by the statement, evolution is not progressive. There's no linear direction in which all branches are going. There are various kinds of evidence for evolution or for documenting the changes in species through time. And of course, the fossil record is tremendously important in this respect. The fossil record is incomplete because not all organisms leave traces in a fossil record, and because of the geological activity of the surface of the Earth, then the edges of plates that are converging, for example, are lost as they're remelted in the core of the Earth. But for the fossil record that we have, it was pretty clear in the 1800s, which is when radioactive dating methods were in fact developed, that the age of the Earth was much older than had been previously appreciated, that the age of the Earth is about 5 billion years, and we will consider later what important changes in life, in the history of life, occurred during those 5 billion years. But at least from the fossil record, it's very clear that there have been many different changes throughout time, that there were organisms that are seen in the fossil record, for example, that have apparently gone extinct because they are no longer found anywhere on the planet. There is also the existence of transitional features seen in some fossils which are intermediate between earlier and later life forms. There's also the existence of so-called vestigial traits, which are very reduced forms of traits which are less reduced in earlier life forms. by the way, a fossil just simply refers to any kind of trace of an organism that lived in the past. So here are some examples in this diagram in front of you right now of different kinds of fossils. Some fossils look more like the organism that had been living in the past and was subsequently, its tissues was subsequently mineralized, such as shown here in the figure on the left. But in the figure on the right, which shows a couple of what look like footprints, these are also fossils because they're traces of an organism, even though it's not a mineralized tissue. And this is an example of fossilized fecal material, which can also be a helpful indicator of organisms that have lived in the past. But let's consider this is an interesting example of extinction. Back in the 1800s, then there was less appreciation for the fact that some organisms had in fact gone extinct over the years, and that is why the Irish elk is such a famous example, because when fossils were found for this particular organism, for this species, which is no longer on the planet, it was such a large and obvious fossil that was described that it was pretty clear that if this animal in fact was still running around somewhere on the planet, that somewhere somebody would have seen it. By comparison, here's a human for a size comparison there. Back to the topic of vestigial traits. This is considered evidence for changes over time, because when one compares traits in closely related individuals, such as humans and other primates, then one can see examples of particular traits which are reduced in humans. So for example, although most primates have tails, which are very important for balance and locomotion in the treetops, we humans that no longer, that don't live in treetops then have a very reduced tails. And in fact, we just have a tiny, tiny little remnant of a tail, which is a bone called the coccyx, and the spelling is shown right here. And this is considered a vestigial trait because it has basically no or reduced function at the current time in our particular species, although it is clearly related fairly directly to functioning structures in other closely related species, meaning in other primates. Another interesting example of vestigial trait are goosebumps. So this is something that one can see in humans, as shown in here on the right. Goosebumps are something that one sees, for example, if a human is chilly, then the little tiny muscles associated with the bases of the hairs contracts, making the hairs more erect. We're not a terribly hairy species compared to our primate relatives, and so this is not quite as impressive a visual display as is seen in primates in the picture shown on the right, for example, where as a chimp erects its hair, it really fluffs out hair all over its entire body, and this is then a very large visual display. So, in order to allow you to self-evaluate if you understand what a vestigial trait is, consider this as a virtual clicker question. Here's the question. Why are some traits described as vestigial? A, they improve the fitness of an individual who bears them compared with the fitness of individuals without those traits. B, they change in response to environmental influences. C, they existed long ago. Or D, they're reduced in size, complexity, and function compared with traits in related species. Consider these four different possibilities here and consider which one best describes a trait that would be defined as or described as a vestigial trait. So I recommend you pause the movie right now and pick your answer. Welcome back. I hope you considered these and picked your favorite answer, and hopefully you picked D as the best answer for this particular question then. The vestigial traits can be identified because they are reduced in size, complexity, and function compared with traits in related species. Another source of evidence that species are related that is closely related to vestigial traits, but considers traits that are not necessarily vestigial but can also still be completely functioning, are traits that are homologies. And homologies are characteristics of organisms that are similar because of shared ancestry. So, for example, if you look at figure 25.9 from the book, which is shown in front of you right now, although the limbs of turtles and humans and horses and birds and bats and seals all are used in very different ways and have many different characteristics, there are certain similarities between these different limbs which are interpreted as resulting from the shared ancestry, namely that all of these limbs are variations on a theme that have evolved then from a common ancestor of all of these groups. And so one can identify the homologous bones between all these different limbs. Homologies don't have to be structural. Homologies can also be developmental. So, for example, if one looks early in development of related species, in this particular figure, humans are contrasted with an immature developing bird and also another immature developing mammal, one can see that if one looks early enough in development, there are certain characteristic traits that are shared between humans and cats and birds, namely tails and gill pouches that are lost in adult humans, but are shared characteristics. And again, they're described as homologies because these are similarities that are shared between closely related species because of shared ancestry. They're derived from modifications on the same traits over time. And then finally, another example of homology would be genetic homologies. This is another example of similarity in genetic sequences that is caused by a shared history, and that the ancestor, for example, of humans and fruit flies presumably had a sequence from which these two were derived later in time.